All right, thanks, Hillary. Just to double check that you can hear me and see my title slide okay.
Yes, we can.
Awesome. Well, thanks for the invite to present to you. I was just checking out um, the list of attendees here, and there's not very many familiar faces, and that is great. It's hopefully this will be some new information to you. And my goal is to just give you an overview of what we're up to here in Minnesota on the aquatic invasive species research front. I'm going to go very fast. So if you have questions, I'll try to save time at the end to get to those afterwards. Otherwise, I'll have my contact information at the end and you can follow up later. I should also point out, given that there's some terrestrial folks in the crowd too, that we do have a sister center at the University of Minnesota that focuses on terrestrial invasive plants and pests. So I'll present on the aquatic side, and if you're curious to know more on the terrestrial half, I can connect you with those people. Okay, so I don't need to belabor any points here that aquatic invasive species are a problem. Minnesota, like New York, has a rich culture in our aquatic environment. Land of 10,000 lakes are something worth protecting. So in 2012, the state of Minnesota basically said enough is enough. The AIS were spreading very quickly, more things were coming in, and we created a center, a research center here at the University of Minnesota to try to fight back. Since then, we've expanded our focus and our capacity, working on many different things. Just briefly, our obligatory mission statement here, just so you understand, we're here to develop research-based solutions to reduce impacts of AIS in Minnesota, and really importantly, to advance knowledge to inspire action by others. We are a bit of an atypical academic program in that we spend a lot of time with that science translation component and try to get the published papers out into the real world. This slide is a little dated now. We have about 30 research fellows affiliated with our program. Those are PhD level scientists, primarily at the University of Minnesota in the Twin Cities, but we have folks up in Duluth and other universities across the state, and research staff at the USGS facility down in La Crosse, Wisconsin. We have 16 incredible graduate students. We have a couple different advisory boards that keep us focused on our mission and the most important work that needs to be done. And like I said, we do a lot of outreach, and we have a citizen science program called AIS Detectors, and we have some internal staff that focus on research outreach and communications. I don't have time to explain our process for doing our work, but I can come back to that in the Q&A if folks are curious. But a long story short, we are very solutions oriented and incorporate perspectives from stakeholders in our process of prioritizing research needs. So given that there's so many problems out there and so many things that could be done, we've created this funnel that goes from all of that possible work down to a fairly focused list of very specific needs that should be pursued. That ultimately gets released through a competitive RFP, and MAISRC supports those projects, and then ultimately, hopefully, those result in real-world solutions. Just to note for the researchers in the room, we do have an RFP that's open right now for about $1.8 million. I'll drop that link when I'm done talking in the chat, just so you have it, but it does require a Minnesota-based PI, but we do encourage collaborations across the country and around the world if you're interested in teaming up with a Minnesota person to work on an AIS topic. We've done a lot of work. We've had in our life about 60 or so research projects. We have 20 going on right now. Those are typically 2-year projects. So a lot of work across organismal groups and approaches. There's been a particular emphasis in the last couple of years to increase our effort in social science research. So that is a growing area right now. On our website that you see listed here, you can find all of this information. We spend a fair bit of time trying to organize research updates in a way that managers and folks in the field can understand. So I encourage you to check that out if you're curious to learn more. But I'm going to touch on just some highlights really quickly, just to give you a sense of the breadth and depth that we're pursuing here at MAISRC. And I'll spend the most time focusing on zebra mussels just as one example of what we're doing, but similar lines of research exist for other species as well. Zebra mussels, like in New York, are a huge problem here in the States. They've invaded about 300 of our inland lakes and rivers. Huge management focus. We've worked to understand the impacts. And of course, we know that there are ecological impacts of the species, but quantifying them in a way that can be useful for management decisions has been really important. So when thinking about recreational fishing, which is a huge economic driver here in the state, we've recently confirmed those impacts to be quite important in lakes that are invaded with zebra mussels, and even more impactful when they're coupled with spiny water fleas. The fish can be about 25% smaller in their first year of life, which plays out throughout the rest of their life. Something that has been increasingly, it's growing on everybody's radar, is the potential impact to human health. And there was a really important paper published a few years ago in Lake Michigan that showed that mercury dynamics within a lake are changing as the food web changes because of invasive zebra and quagga mussels. So we've just wrapping up a project that looked at mercury concentrations in walleye and yellow perch in invaded and uninvaded lakes. The student defended her thesis just yesterday, and so I don't want to, the paper will be forthcoming, but I'll just summarize by saying that the impacts are significant. The walleye in invaded lakes, mercury concentrations were well above the safe consumption advisories set by our Department of Health compared to uninvaded lakes. So this deserves much more research. And if you're not tracking the human health implications of this species, I encourage you to do so. We've looked at spread as an important aspect of defining and informing prevention activities. We've looked at spread using genetic markers, essentially the ancestry.com of the zebra mussel family tree here in Minnesota. We've looked at more quantitative ways of just looking at boats, at peligers and adults to see how they're spreading and what that risk might be. We've looked at finding mussels in a variety of different ways. We've standardized survey methods that are being used by our local and state biologists now. We have a fairly large project at this point that is looking at standardizing methods for eDNA detection of zebra mussels and other species. And we're wrapping up a 5-year effort right now that has been developing a multi-beam sonar technology to map zebra mussel populations within a lake and river system. We developed an online tool called the AIS Explorer that predicts the likelihood of zebra mussel establishment. And this is there for starry stonewort as well. So at the lake level, what's the likelihood that the species will get there and survive? This is really important, of course, for surveillance activities when we have so many lakes to choose from. So the underlying data has also been useful for optimization models to locate watercraft inspectors. So how can we be most efficient with the limited resources we have? These models have been used by many of our counties to inform their programs. For established populations, we've been scaling up control efforts. So it's not, I don't think, something that is inevitable that once a lake gets zebra mussels that will always be there. I think there could be technologies off the shelf maybe, or could be developed to suppress or maybe even eradicate them. And that's a research goal. So we've been working on this for quite a few years now. We had the largest ever treatment 2 summers ago in Lake Minnetonka near the Twin Cities. We've been monitoring that population. The multi-year suppression has been very successful. And this project is going to be continuing these next few years, refining strategies and hopefully being deployed as a management tool soon. We also sequenced the zebra mussel genome recently. This was a big breakthrough for many reasons, but primarily, I mean, I don't think we can rely on copper as a control strategy, and something that is more species specific ought to be pursued as well. So we currently have a large project looking at the development of RNAi. So using RNA interference to essentially manage and control or eradicate zebra mussels. And we just got a sneak peek of some of those data yesterday. They're not ready to share it yet, but it's very exciting. And this is something that people should keep an eye on. So shifting gears a little bit to invasive plants, some of the same problematic actors as you have in New York, Eurasian water milfoil and the hybrid variety, which is becoming more of a problem in the Great Lakes region, curly-leaf pondweed, and other starry stonewort, of course. One that we've made the biggest splash recently has been our research efforts on non-native Phragmites. We had a series of projects that have been used to inform a statewide management plan that we wrote and is now starting to be implemented across the state. The bad news here in Minnesota is we have about 400 populations of Phragmites across the state. But the good news is that they're small in their sort of invasion. So we think that there are opportunities for very strategic rapid response efforts to eradicate this species across the landscape and manage the larger, more established populations to prevent further spread. So this is one that I suspect will be one of those success stories in the coming years, thanks to our research program and the state's commitment to doing something about it. Here in Minnesota, we have the big-headed carp swimming up the Mississippi River. The formal invasion front is down in Iowa, but they are starting to trickle in. We capture bighead and sliver carp every year through commercial fishing. We've been spending a fair time looking at deterrent strategies at the lock and dam structures to slow them down. And that work continues. A species that we've made the most progress on for the fish side, though, has been the common carp, arguably the country's most ecologically damaging aquatic invasive species. And we've thrown everything, including the kitchen sink, at this species, trying to figure out ways to control it, suppress populations, prevent reproduction, all of that stuff. And through more traditional integrated pest management strategies like barriers and netting and predatory biocontrol of the carp eggs, those have all been successful, but we also have more innovative control strategies, including genetic modification of common carp and pathogenic biocontrol using the koi herpes virus that are currently being investigated, which is hopefully exciting news to come on both of those fronts. We've also, just to show you where this research can ultimately go, we've spun off a company to implement a lot of these management strategies in the real world. Community engagement's key to doing the research and also implementing the research. So we spend, again, a lot of time on this. We're very proud of our citizen science program in partnership with the University of Minnesota's Extension program. The AIS Detectors is what it's called. We have 300 certified AIS detectors across the state. They have a variety of activities that they can participate in. They do a lot of local volunteer service as part of that certification. Their impact cannot be understated. It's been significant. For example, we have this Starry Trek event every August where these trained volunteers go out and look for starry stonewort. They've found in Minnesota about 1/3 of the known infestations through, these are just regular people going out and looking for it, and it's been critical to the state's response for that species. So that's a very fast overview of what we've just touching on some of the things that we've been doing. We've accomplished a lot. We're pretty proud of what we've been able to do with the state's support. but acknowledge that we have a long ways to go to really solving these problems. And so I hope that I can come back someday, or you'll stay tuned to see more of this work progress. But the take-home message that keeps us excited are starting to see research being implemented by managers. And these stories we can start to tell now. We're 8-9 years into our life, and we're starting to see local, state level managers implementing these results and seeing meaningful impacts across the landscape. And that is really rewarding for us on the research side, and really important for the public resource. If you're curious to know more, hopefully I've saved time for some Q&A, but there are lots of ways to stay updated on the work that we're doing. And although we're focused here on Minnesota, our study sites are largely here within this state, the species that we're working on and the approaches we're using are likely applicable in New York and elsewhere. So I encourage you to check out our website. If you want to sign up for our newsletter, we send one out every couple months. That's the easiest way to stay updated on research highlights and events. We're fairly active on social media if you're into that. We also have a Detectors webinar series. It's primarily focused on a public and management audience. It's not intended for researchers, so it's presented in a different way than a normal scientific conference. Our next webinar is going to be tomorrow, actually, if you're interested in narrowleaf cattail, one of our projects focusing on that species, presenting an update on their work. We have an annual research and management showcase. It's our conference where we present all of our research findings. It will be on September 21st of this year. It's going to be online. We've had to shift because of the pandemic, and it turns out that that has been a win-win for everybody. It's been good for us, and it's also really increased attendance from across Minnesota, across the region. I know there have been folks from New York that have participated in that conference virtually the last couple of years. And if you have any questions, don't ever hesitate to just email us, email me, or one of the researchers, and we can help get your questions answered. So with that, I'll thank you again for the invitation. And if we've got time, Hillary, for Q&A, we can do that as well.